is Sina, and today I'm going to talk to you about annotating sequencing reads with a new machine-readable sequence structure format called seqspec. Now, one thing that's really exciting to me about the field of genomics is the decreasing cost of sequencing, which has led to an explosion in the number of assays, single cell or otherwise genomic assays, and a proportionally large amount of data and more complexity. And so as you can see here, as the cost of the genome has decreased over time, the amount of data on the SRA or the sequence read archive has increased. And a really cool resource called Encyclopedia demonstrates that there is a numerous amount of assays and continues to be. And consortium activities like those in IGVF actually reflect this trend. And so I'm highlighting here just a few of the sequencing-based assays that investigators within IGVF plan on undertaking. And so with such a wide diversity of assays, it begs the question, how are these assays organized? How do they compare to each other? And how can we actually understand the contents of sequencing reads generated from these assays? And so I think one really interesting organizing principle that I've realized throughout the course of my PhD and postdoc is that single-cell genomics assays fall under three main categories, or they can be thought of as occurring in three steps, namely physical isolation, followed by molecular capture, and then lastly by library generation, after which sequencing is performed. Physical isolation can occur in wells, like in SmartSeq v2, in shells, like in the 10x Genomics assay or in DropSeq, or the cell itself can be a container, like SplitSeq. And once containerized, cellular contents are then tagged with synthetic molecules, and various biological modes can be assayed, whether it be RNA, DNA, protein, or whatever. And the sequencing library is generated from this tagging process, whereby sequencing adapters on the 5' and 3' end are attached to an insert to generate a sequencing library for sequencing. And this is just a simple example on the bottom here showing you an example sequencing library for the 10x Genomics v3 assay, which contains a 16 base pair barcode, a 12 base pair UMI, and then cDNA, which is the molecular mode that was captured. I want to focus today on molecule capture and library preparation because those are the key steps for generating sequencing libraries. The three main sequencing companies that have sequencing machines, namely Illumina, PacBio, and Oxford Nanopore, are broadly based on the following steps, namely cutting DNA, performing end repair, and then ligating sequencing adapters. After which sequencing is initiated at the sequencing adapters. So as I showed you on a previous slide, after physical isolation and molecular tagging, you get this kind of construct here. And just to map it to some real-world example, this is the library structure that 10x Genomics specifies for their multiome assay, but the RNA part of the multiome assay, where they have the 5' sequencing adapter on the left, the 3' sequencing adapter on the right, and then the custom insert that's designed by the assay, namely a cellular barcode, a molecular barcode, the UMI, and then poly-T stretch followed by sequencing reads that assay the cDNA. And so documenting library structure is identifying adapters and specifying the insert design. An interesting observation is that sequencing libraries are comprised of these atomic regions, things like cell barcodes, unique molecular identifiers, and genomic features like cDNA or genomic DNA. And what's really cool is that sequencing libraries are like Lego blocks. So you can build a sequencing library by selecting the appropriate 5' sequencing adapter, the 3' sequencing adapter, and designing a custom insert within the middle based off your assay of choice, where you can assay DNA like in whole genome sequencing or in chromatin accessibility, or you can assay RNA like in single-cell RNA-seq. You can assay proteins, cell surface proteins like in CiteSeq, or you can have a multiome or multimodal extension where you assay many at the same time. So we came up with a new file format specification as well as command line tool called seqspec. Now, seqspec is a file format, which looks like down here, based off YAML, that allows users to identify the elements of a sequencing read and annotate them. So elements in the assay can be annotated, namely, what is the sequencing machine that was used to generate the data? What is the specific assay that was used? What are the modes or the multimodality of the assay? Namely, are you assaying RNA? Are you assaying chromatin accessibility or some combination of them? And a key feature of seqspec is that atomic regions can be annotated within your sequencing reads by saying this stretch of a read corresponds to a barcode or this stretch of a read corresponds to UMI. And then sets of those regions can be ascribed the tag of a meta region. And often I've found it to be useful to associate these with the names of FASTQ files that contain those elements. And then the modality itself can comprise the entirety of this annotation. So if you think of this as a sequencing read, we can annotate the elements of that sequencing read in multiple levels. And this creates a kind of graph structure or kind of ordered tree structure, which we can then represent as a tree and then ultimately as a file format, as a YAML file. Now, seqspec is cool. As I mentioned before, it's based off the YAML file format, so it makes it quite easy to write by humans. And it's also machine readable, which makes it compatible with the seqspec command line tool, which we also developed. You can annotate assays as well as regions. Regions can be nested within regions. The specification is modality agnostic, meaning we can specify multiple kinds of modes. It's also region agnostic, so whether the region that we're identifying is a fixed length cellular barcode or a variable length region like genomic features, or if it is a region that is derived from some kind of on list, or if it's a fixed region. It's agnostic in that way. The specification is compatible with the command line tool, the seqspec command line tool, which allows us to auto-check the formatting of the specification as well as perform various operations on it. And it's quite straightforward, actually, to initialize a new specification for a new assay that you developed using this Newick format, so a string called a Newick format, which is often used for specifying phylogenetic trees. And so we've put together a preprint that specifies the seqspec format as well as the tool, and we have a code base that you can check out to learn how to make them as well as understand more about the specification. So in summary, single-cell genomics assays perform physical encapsulation in wells, shells, or cells, followed by molecular barcoding and library preparation. Sequencing libraries are comprised of common atomic regions that we can specify with the seqspec file format. The seqspec is a file format specification based on YAML that allows users to specify the elements of a sequencing library, annotate regions with metadata, such as fixed sequences or variable length sequences, and then associate regions with FASTQ files. This makes it really convenient for assay developers and tool developers to have a lingua franca, or a means of communicating between each other through this specification, so that tool developers know which parts of the sequencing reads are important for performing downstream preprocessing. seqspec is also a command line tool for manipulating seqspec files that can auto-format a specification and check for the correctness against a spec, that can automatically extract positional information, for example, for barcode, UMI, or genomic feature elements. It can automatically identify and build on lists for barcodes, and seqspecs can be quite easily initialized, or it's quite straightforward to initialize a seqspec using this Newick format that I mentioned earlier. So for more information, please check out the GitHub repository. And with that, I'd like to acknowledge the folks at Caltech, the folks at UCI, University of Iceland, and those in IGVF. Thank you.